Welcome friends. Today we'll be discussing metabolism of aliphatic amino acids. Among these, we are discussing very important amino acid, that is glycine, which is important for you for students. So, metabolism is of two important processes: catabolism and anabolism. So, catabolism is breakdown, and anabolism is synthesis. So as you know, glycine is non-essential amino acid, that is, it is synthesized in our body. What are essential amino acids? Essential amino acids are the amino acids which are not synthesized in our body, but they have to be supplied from diet. So methionine, threonine, valine, leucine, isoleucine, phenylalanine, tryptophan, these are called essential amino acids. They are not synthesized in our body. Histidine and arginine, they are semi-essential amino acids. Excluding these, all are non-essential amino acids. So glycine is non-essential amino acids. That means it is synthesized in our body. So the first reaction by which glycine is synthesized is from serine. Serine is converted into glycine in presence of enzyme serine hydroxymethyl transferase. So here tetrahydrofolic acid is converted into N5N10 methylene tetrahydrofolic acid. Next important compound which gives rise to glycine is threonine. So threonine in presence of enzyme threonine aldolase is converted into glycine and acetaldehyde. So from threonine, glycine is formed. Third most important reaction, which is reversible, is carbon dioxide combines with ammonia in presence of enzyme glycine synthase complex. It forms glycine. So how glycine is catabolized? So glycine is catabolized. The first important reaction by which glycine is catabolized is in presence of enzyme glycine decarboxylase, pyridoxal phosphate. It acts as coenzyme here, pyridoxal phosphate. It is pyridoxine is the vitamin B12, B complex vitamin pyridoxine, and its coenzyme form is pyridoxal phosphate. So decarboxylation reaction occurs here. Glycine is converted into amino-methyl group in presence of pyridoxal phosphate. So this amino-methyl group, it gives rise to ammonia. So tetrahydrofolic acid is converted into methylene tetrahydrofolic acid. Lipomide is converted into reduced lipomide, and NADH2 is converted into NAD. So here glycine is ultimately converted into carbon dioxide and ammonia. So glycine's cleavage system, glycine undergoes oxidative deamination to form ammonia, carbon dioxide, and one carbon unit, that is methylene tetrahydrofolic acid. You can see here carbon dioxide is formed, ammonia is formed, and one carbon unit, methylene tetrahydrofolic acid. The glycine cleavage system is multi-enzyme complex consisting of glycine decarboxylase, amino-methyl transferase, methylene tetrahydrofolate synthase, and NAD-dependent lipomide dehydrogenase. So deamination of serine to pyruvate. Serine is converted into, say, in presence of serine dehydratase, pyruvate. So from serine, glycine is formed. Serine is converted into glycine, serine hydroxymethyl transferase. So you see here overview of glycine metabolism. So you can see from carbon dioxide, ammonia, and one carbon unit, glycine is formed. From proteins breakdown, glycine is formed. From serine, glycine is formed. From threonine, glycine is formed. So we can say these are the anabolic reactions by which glycine is formed in our body. So which are the catabolic reactions? How glycine is catabolized? So glycine is converted into ammonia and carbon dioxide and one carbon unit. Ammonia is ultimately converted into urea. Glycine is converted into serine because this is reversible reaction. Serine is converted into glycine, glycine is converted into serine, and serine is ultimately converted into pyruvate, which is converted into glucose. Serine can be converted into cysteine, serine can be converted into choline. Then glycine is converted into glycolic acid, which is ultimately converted into oxalates. So glycine is converted into creatine. That is very important. So glycine combines with arginine to form guanidoacetate. In presence of enzyme glycine arginine transamidinase. Here amino group is transferred. So guanidoacetate combines with S-adenosylmethionine. So methyl group is donated by S-adenosylmethionine. So guanidoacetate combines with that methyl group to form creatine, and creatine is converted into creatine phosphate. And this creatine phosphate is non-enzymatically anhydration occurs of creatine phosphate is converted into creatinine, which is excreted into urine. Glycine forms C4, C5 and N7. It is nitrogen atom number 7 of purine ring. So carbon number 4, carbon atom number 4, carbon atom number 5 and nitrogen atom number 7 of purine ring is contributed by glycine. Next important function of glycine is glycine combines with succinyl-CoA to form delta aminolevulinic acid ALA, and which ultimately gives rise to heme. Then glycine combines with cysteine, which combines with glutamic acid to form tripeptide glutathione. Then glycine forms bile salts. So glycine plus cholyl-CoA glycolic acid, which combines with sodium to form sodium glycocholate. Then glycine combines with benzoic acid to form hippuric acid. This is called conjugation reaction. So we'll see one by one metabolic functions of glycine. So glycine may be used for biosynthesis of following compounds: creatine, creatine phosphate, creatinine. Second, heme. Third, purine nucleotides. Fourth, glutathione. Fifth, it acts as inhibitor neurotransmitter. Next, it converts into serine. Then conjugation of bile acids. Then detoxification of benzoate to form hippurate. Then it contributes to one carbon pool, glycine synthesis system, glycine cleavage system, and it acts as it is glucogenic because pyruvate is converted into glucose. So these are metabolic functions of glycine. So I have already told you how creatine form from glycine. So glycine plus arginine, step 1, in presence of glycine arginine amide transferase, it forms ornithine and guanidoacetate. Guanidoacetate takes methyl group from S-adenosylmethionine in presence of guanidoacetate methyl transferase to form creatine. Here SAM is converted into SAH. Creatine in presence of creatine combines with ATP in presence of creatine kinase to form creatine phosphate. ADP is formed, and this creatine phosphate spontaneously non-enzymatic removal of water and phosphate group creatine is formed. This creatine is excreted in urine. So in muscular dystrophy, creatine kinase MM is present. In brain, creatine kinase BB isoenzyme is present. In heart, creatine kinase MB isoenzyme is present. So CKMB is increased in myocardial infarction. CKMB is increased in muscular dystrophy. Creatine is converted to creatinine, and this creatine phosphate is stored in the form of energy in muscles. Creatine urinary creatine excretion normal, it is negligible. Increase in muscular dystrophy, muscle injuries. Normal serum level is 0.2 to 0.4 to 4 milligram per deciliter, and increased in muscular dystrophy. Serum creatine in normal range is 0.7 to 1.4 milligram per deciliter. In males, 0.6 to 1.3 milligram per deciliter for females. Increased in renal failure. Serum creatine level is increased in renal failure. It is better index than blood urea. Creatinine clearance test for glomerular filtration test is very important. Creatinine production is spontaneous, continuous, no fluctuation is there, and depends on muscle mass. creatine excretion is constant for a particular person. 24-hour urine sample is done, creatine clearance test, urinary concentration per gram of creatine. So glycine plus arginine, guanidine acetate. This step occurs in kidney. Guanidine acetate takes methyl group to form creatine. This step occurs in liver. Creatine combines with ATP in presence of creatine kinase to form creatine phosphate. This step occurs in muscles. Creatine phosphate spontaneously converts into creatinine. Again, this step occurs in muscle. So clinical application of creatine and creatine, normal range I have told you, 0.7 to 1.4 milligram percent. It is sensitive indicator of renal function or kidney function tests. Blood creatine levels are increased in kidney disease. In early phase of muscular dystrophies, blood creatine level and are increased, and urinary creatine levels are also increased. Apart from muscular dystrophies, reduced muscle mass is also cause of low creatinine in women, old age, bedridden states, and low protein diet. CKMB is increased in myocardial infarction. So special metabolic functions: biosynthesis of heme, creatine, purine nucleotides, glutathione used as conjugating agent. Conjugates with bile acids, glycolic acid, glycosaminoglycolic acid. Conjugates with benzoic acid to form hippuric acid, which is excreted in urine. It acts as neurotransmitter in brainstem and spinal cord. Very high levels causes overexcitation. It is constituent of proteins seen at bends or turns in collagen. Every third amino acid is glycine. It forms most important compound in our body, heme, glycine plus succinyl-CoA. In presence of ALA synthase, it forms delta ALA, which forms heme. So that's all. We'll discuss disorders due to defective glycine metabolism in next topic.